protein hormones, and that is essentially a historical, not artifact, but a historical arrangement that came about because the only thing you have is a biological source for the material, the better you are at purifying the material, the closer you are to a pure hormone, the better that hormone will work in any bioassay system. So they came to the conclusion that the way to measure it was to decide at some point what one unit of activity was, bioactivity was, and to adopt a standard preparation that was so many international units of activity per microgram or milligram of substance. That was peculiar to each hormone. So insulin international units are not the same as parathyroid hormone international units, which are not the same as follicle stimulating international units, etc. In terms of pure hormone preparations, which are now more available because of the cloning of the genes that make up the subunits or the hormones themselves, or the basis for the hormones themselves, you can essentially put a number of international units on a given number of nanograms or micrograms of hormone. And when it comes down to LH, for instance, or chorionic gonadotropin, they're both pretty close. They're around 10 to 13,000 international units per milligram of protein. So that's kind of where the translation comes into. So we've spoken a lot about the gonadotropins. I want to spend a little bit of time now talking about the growth hormone axis and the proteins of the growth hormone axis and their relationship to prolactin. Because it turns out that growth hormone, prolactin, and a placental hormone called human choreo mammotropin, or HCS, which also is known as human placental lactogen, HPL, are all genetically, structurally related to one another. They are single chain proteins. These are all genetic structural relatives. They're one chain proteins, protein hormones. And they're coded by a cluster of genes. In the human, it's on chromosome 17 on the quiniquine straining arm in position 22 to 24. And there are 1, 2, 3, 4, 5. Let's see, one of these is a pseudogene, but the rest of them are functional. So there's multiple placental lactogen genes and a couple of genes for growth hormone. The prolactin gene is not too far away from this. Okay. Each gene has 5 exons that go on to code the final protein MHF4 introns. So they're definitely genetically related to one another. The prolactin gene is the same structure. Okay. Growth hormone and prolactin, if given in adequate quantities, will stimulate each other's receptors. I think I spoke about that. People with acromegaly can have galactorrhea, and in the male it's obviously abnormal. And those people who have prolactinomas often have effects that are associated with growth hormone, normally associated with growth hormone overproduction. So you can get diabetic-like conditions, for instance, with hyperprolactinemia. So you've got hormones that act via somewhat similar receptors in different tissues. Their structures are similar. Their genes are obviously similar. Now, one thing I didn't point out with the gonadotropins and HCG, but I need to make that point here. It also applies to that group. You know about the feedback loops here. So there's homeostatic control controls on these. Okay? And you need to know what those loops look like. For the placental hormones, there's no homeostatic control. So for some reason, there's no apparent feedback controlling how much of those things are made. Now, when you get to the growth hormones, we talk about the feedback loop and the fact that in the growth hormone particularly, part of this feedback loop controlling growth hormone, releasing hormone secretion in the hypothalamus has to do with the production at end organs, somatic tissues, of metabolites, things like amino acids, fatty acids, glucose, that end up being sensed in the central nervous system and providing the negative feedback onto this system. Prolactin is the man out on the anterior pituitary in having a negative major controller in the form of dopamine that tends to suppress its production. Okay, so the other thing that this particular group of hormones is noted for is, I will hope, shown in this other slide. Okay, so I want to go, let's see if I got that. Yeah, that's the slide. Okay. As you may remember, growth hormone, in acting at its end tissues, stimulates the production of insulin-like growth factors. So IGF-1 and IGF-2 here. Well, it turns out that there are modulators of the concentration, the local concentration, and the circulating concentration of IGF-1 and 2 in the form of a host of binding proteins. There are 6 of them known. So 6 insulin-like growth factor binding proteins. Okay. And those insulin-like growth factor binding proteins will tend to modulate the concentration of these in any particular tissue. So it's a balancing act. It's, yes, hormone binding to receptor, but it's not just one binding protein, but there may be several. These aren't, all 6 aren't made everywhere. And they're not made all the time. So tissues may have selective production of subgroups of the binding proteins. So there's now a whole body of literature trying to describe when these are produced, in what quantities, to buffer the concentration of free, biologically available active hormone in any particular situation. So binding proteins, as you know, can drain off free hormone and temper the amount that's available to bind to a particular receptor. But the situation is even more interesting than that, in that there are particular proteases that are available that may cleave the binding proteins. This gets to be very interesting in the case of a trophoblast, a fertilized early stage embryo trying to implant into the wall of the uterus, because the trophoblast generates insulin-like growth factor 1. The maternal epithelium, which is the surface of the endometrium where this embryo is trying to implant, produces IGF binding protein. If I'm not mistaken, it's binding protein 4 most commonly. So it's decreasing the amount of IGF-1 that's available to bind to receptors, where the receptors would normally be both on the embryo and on the maternal epithelium, where in either case it would stimulate growth. So embryo makes that, mom makes that, but then the embryo makes an IGF binding protein, protease, that cleaves this and makes this more available. But then mom makes an inhibitor for that protease. Okay. So there's a conversation that goes on to optimize the timing and positioning of the site of implantation for that particular trophoblast. And there are other situations where you could see this kind of a conversation going on, at least locally, to optimize the actions of this hormone, and by implication, the growth hormone that produced it, so that tissues are responding optimally under appropriate conditions. So conversation, it's not as simple as a hormone simply binding to a receptor. All right. In addition, there is the growth hormone receptor comes in two forms, a small form and a large form. The large form has a larger extracellular domain. And that extracellular domain can be cleaved at the cell surface, which makes that extracellular domain available in circulation as a growth hormone binding protein. So you've got another buffering system, not dealing with growth hormone, insulin-like growth factors, but with growth hormone itself. So we've got, even with these protein hormones, we have complicated systems that help modulate their actions at a given time in particular tissues.
I'm sorry, could you just repeat what you said about the domain and how it acts with the
Oh, growth hormone receptor, two forms, small and large. The large form, the extracellular domain, can be cleaved at the surface of the cell, which frees that domain to circulate, and it can bind hormone. So it effectively becomes a growth hormone binding protein. Not a insulin-like growth factor binding protein, but actual growth hormone binding protein. Okay. And you probably will see some conversation of that in the text at this point. It's taken a while for this area to develop, but the conversation between mom and embedding embryo is a very interesting one that we've come to understand better now. Okay, so that's kind of where I want to leave it for growth hormone, and prolactin gene products. We'll come back to talk about prolactin actions in lactation when we get to that topic a bit later in the term. And the other thing I wanted to go back to is pro-opiomelanocortin. I drew this briefly on the board a few days back. Pro-opiomelanocortin is this 35,000 molecular weight protein translation product that is gradually cleaved post-translationally in the cells that make it. So the cells that make this stuff are some of the neurons in the central nervous system, the corticotropes in the anterior pituitary, and the melanotropes that exist in the intermediate lobe of the pituitary. So pro-opiomelanocortin gets cleaved initially into two pieces, pro-ACTH and beta-lipotropin. Beta-lipotropin has some effect on fatty acid metabolism, but it's not really a primary controller even of that metabolism. Pro-ACTH continues to be cleaved to additional fragments. There's a N-terminal 16K fragment, and there's a 39 amino acid long peptide or small protein called adrenocorticotropic hormone, which you've already learned about. That's involved where? What does that hormone do? What does ACTH do? What does it do to glucocorticoids? Pardon? It stimulates release from where? From where in the adrenal glands? The cortex, yes. Can you be more specific than the cortex? There are two layers, yeah. It comes from the corticotrope. It acts on the adrenal cortex to stimulate glucocorticoid production in two layers of the adrenal cortex. Those two layers are what? The fasciculata and the reticularis. Okay. Fasciculata, reticularis, and the outermost layer, which is not controlled by ACTH, is the what?
Of the adrenal cortex?
Yeah. It's called the glomerular layer, or the glomerulus. Yeah, it's the outermost layer of the adrenal cortex. Okay.
What? Say that word again.
Actually, let me do something here. Okay. Here's the adrenal gland. The middle part, the medulla, is a modified ganglia. It's a derivative of the nervous system. It produces epinephrine and norepinephrine. The next layer out is the reticularis. It produces primarily androgens. The next layer out is the fasciculata. That's the one that's producing primarily the glucocorticoids, depending on which species you happen to be. You're either producing cortisol, like you'd see in humans, other primates, sheep, or corticosterone if you're a rat. Okay. And then there's this outermost layer, the glomerulosa layer. And the glomerulosa layer you can best keep in mind because the product is this aldosterone, which is kind of an end of the steroid metabolic pathway, and it affects sodium reuptake in the kidney. Kidneys are, the functional portion of the kidney is the glomerulus, okay? So this is the part that affects the kidney. This is not under ACTH feedback control. This is not under ACTH feedback control. But these two layers are. Okay. So. Back. So that's going on fairly rapidly, and the breakdown is going on fairly rapidly in the corticotrope, particularly, and it gets to essentially this level in the corticotrope. So you've got ACTH, you have a protein called gamma-lipotropin that is also involved in controlling fat metabolism. And then you've got this beast of 31 amino acids out here at the end called beta endorphin. What is endorphin? Any runners here? Distance swimmers? No, it's not adrenaline.
It makes you happy.
It actually is synergistic with some of the actions with adrenaline.
Happy feeling after you're done exercising.
Okay, the happy feeling after you're done exercising.
Doesn't it have to do with pain too?
What does it do with pain?
It's synthetic, it's natural opiates, right? So it actually suppresses.
Okay, it is an endogenous or natural opiate. It suppresses pain. Okay. Beta endorphin, you want to see high levels of beta endorphin, get a blood sample from a woman in labor. Okay. Her endorphins are sky high. Someone who is responding to a traumatic situation often has endorphins that are really high. You've heard of little people lifting cars off of other people, things that seem to be almost superhuman. What you're usually seeing is a combination of the actions of adrenaline and endorphins. Yeah.
With dopamine, you also be similar function?
No.
No? But dopamine also triggers.
Dopamine, one of the dopamine circuits, remember there are 10 of them, okay, in the nervous system. At least one of them is associated with pleasure, okay? So dopamine levels are high in those people who've just engaged in intercourse, okay, most of the time. Or, you know, after a very successful situation in, say, a competition, those levels would tend to be high. Okay, but let me come back to here. So endogenous opiates, and indeed, opiate receptors, there are multiple opiate receptors, and they will bind beta endorphin, and a variety of other metabolites called enkephalins. Now, if you are looking at a melanotrope in the intermediate lobe, you'll see these fragments being broken down further to various forms of MSH, melanocyte-stimulating hormone, and this beast called corticotropin-like intermediate lobe peptide, CLIP. So this is the end of metabolism for the melanotrope. This is the end of metabolism for a corticotrope. Okay. The endorphins often are also related to enkephalins. There's an alpha endorphin, and there are various other forms that you find that will bind preferentially to some of the receptors, the opiate receptors. Okay. So, you need to know that breakdown occurs. This breakdown is what's called post-translational actions or post-translational cleavages occurring in a protein hormone. I did not want to leave you with the impression that all these protein hormones are simply pieced together by processes other than the processes that are normally used to make proteins. So I want to come back and make sure that you understand how proteins are normally made. So if I had to draw a picture of it, how are proteins normally made? What do we start with? We start with DNA. Okay. And in eukaryotes, how is the DNA organized?
Double helix.
Pardon?
Double helix.
It's a double helix, but in an area that's going to be made into a protein, what does it look like? It's in the nucleus, definitely. How are genes arranged in eukaryotes? They're coiled, fine.
With histones?
Yeah, they're coiled with histones. But if you had to show the organization of a gene in a eukaryote, how would you draw it? Straight. Okay. All right. So how is it organized? Other than 3' to 5', and as a double helix. Okay. There are antiparallel strands, only one of which is going to code for the product. Okay? So let's concentrate on that one, the coding strand. And how is the information organized in this? Pardon?
A promoter region?
There's a promoter region. Then what? Pardon? There's an initiation site. And then what? Pardon?
Regulatory sequence.
The regulator region is the promoter region. Come on, folks. How's this gene organized? In eukaryotes, you learn this in cell biology, or maybe high school biology. The nucleotides form part of the codons, but how is that overall organized?
Exons, introns.
Okay, somebody said exon. Exons and introns. How are they organized?
Exons and then big spaces of introns, exons.
Okay, so you've got segments of the DNA, some of which are exons, intron, exon, intron, exon, intron. And somewhere down here, you've got a stop codon. So what happens from here? That's the structure of the gene, the coding sequence on the gene. What happens from there? Somebody said RNA.
mRNA?
Not, well, you're
It's
not
not
quite
fully
to
mature.
mRNA.
It's not fully
You're not quite to the
Not
mRNA.
fully mature.
Okay, what do you start with? Okay, a polymerase will sit here with modulating elements like hormone receptors, complexes, any transcription type factors, which sit on the promoter region. Sometimes the promoter regions can also sit on these introns. What goes on next? What's the next step? This is 1. What's 2? Have transcription. What's the first product in transcription?
RNA.
It's an RNA, but what kind of an RNA?
mRNA.
It's not mRNA yet. It's
It's
pre-mRNA. It's called heteronuclear RNA. It's full-length RNA. It starts here, and it goes all the way to here. And it still has in it information that codes for both the exons and the introns. What happens next?
Splice.
Okay, you have splicing. What happens during splicing?
Cut out introns.
Introns are removed.
What's removed?
Introns.
Introns are removed. So if that's the exon, you go there, you get rid of that, and that, and that. So we end up with exon 1, 2, and 3. Then what happens?
Want to get technical modification?
I want you to get technical. I want to know what you know about this.
A 5' cap and a poly A tail.
Okay, you got a 5' cap. And what's over here?
Poly A tails for stimulation.
Okay. So splicing and then capping and tailing. And what's the molecule you end up with? Mature mRNA. What happens next? Translation. Well, where does translation occur?
Cytoplasm.
On ribosomes. Where are most of the ribosomes located? A lot of them are in the cytosol.
Rough endoplasmic.
Where else? Rough endoplasmic reticulum. Okay. So the RER, translation on, is it single ribosomes? Okay. Often multiples, and those are called polyribosomes. And or RER, rough endoplasmic reticulum. What's the smooth endoplasmic reticulum?
Synthesis of
Pardon? There's no ribosomes, but you'll find out there's a concentration of other kinds of enzymes. We'll talk about that more when we get to things like steroidogenesis. Okay, so you get translation, and what's the end result of translation? Protein. Is it just simply protein? Yeah. You get, pardon? You get a polypeptide? You get an amino acid chain, okay? Is the amino acid chain complete at that point?
No.
No.
Yes, the undergo structural change to become active.
Okay. Where is this located in the cell? If it's been made on polysomes that are in the cytosol, it will be in the cytosol. What if it's made on polysomes or ribosomes that are associated with the rough endoplasmic reticulum? What?
Aren't they membrane proteins?
A lot of membrane proteins are made on RER. Any protein that's going to be secreted is also going to be made on the rough endoplasmic reticulum. Are hormones secreted?
Yes.
A lot of them, most of them are. Certainly protein hormones are. So they're going to be made on the rough endoplasmic reticulum. Now, where does this product of translation actually end up being if it's made on the rough endoplasmic reticulum? It'll eventually be in the extracellular space. Hang on, we're not quite to the Golgi yet. Okay. It gets inserted into the lumen of the endoplasmic reticulum, which is topologically, what does topological mean? A relative space, okay? Anybody know what a torus is? What's a torus?
It's a three-dimensional object with one surface.
Three-dimensional object with one surface.
It's n-dimensional.
Yeah, well, it ends up being, if you try and visualize it, it looks like a twisted donut.
Yeah.
Okay, where the outside becomes the inside. Okay. Pardon? Mobius strip is another example of it. The idea is that topology refers to contours and shapes and spatial relationships. Topologically, proteins, surface proteins, proteins that are going to be secreted are in the lumen of the endoplasmic reticulum, so they are topologically as if they were outside the cell. Okay? So they end up in the inside vesicles, inside the lumen of the rough endoplasmic reticulum, and they remain in that relationship until they get to the outside of the cell. Now, so this is going into the lumen of the RER. Is it a straight shot to the membrane from there?
No.
What happens? Pardon? It's what? It's packaged. That comes late, actually. Did you have a comment?
No.
Okay. Okay. What happens to this initial transcript if it's a protein that's been made on the RER? It goes to the Golgi, but only after a couple of other things have happened. Pardon? A modification. These are called post-translational modifications. They start in the rough endoplasmic reticulum. What's the first one? No. Well, it's an action on the primary structure.
Leave the portion that's embedded into the
Okay. When they are making these, there's a segment, usually at this end, that forms an alpha helix. It often has hydrophobic amino acids in it. It's called the leader or signal sequence. And it stays embedded in the membrane of the rough endoplasmic reticulum. It helps pull allow the protein to be pushed through the membrane along with a protein channel. And that leader sequence is clipped off. Does anybody know how long that leader sequence usually is? How long is a leader sequence? Guesses?
Six.
Six amino acids? 15 amino acids? Do I hear 20? Do I hear 30? Do I hear 150? Okay. It's usually not big. It's usually on the order of 15 to 30 amino acids long. 6 is probably too short to make the helix come and actually have it penetrate through the membrane. So it's on the order of 15 to 30 amino acids. There are exceptions. There are some there it's 120 amino acids long. But you've got a leader sequence being clipped. What else is going to occur while it's moving through the rough endoplasmic reticulum? Particularly, it also continues on into the early stages in the Golgi apparatus as it moves through that.
I have a question first.
Sure.
I know that for nuclear proteins, the sequence isn't cleaved, so for hormones that eventually act in the nucleus, are those not cleaved?
Which hormones would be made in that cell to act in that cell's nucleus? Okay. Okay. It's not going to happen. Okay. Receptors may retain the leader sequence because they are anchored in the membrane. Okay. But other things in terms of post-translational modification will occur for those hormones or receptors. What other post-translational modifications have you heard about? Methylation, acetylation, sometimes phosphorylation. Glycosylation actually occurs primarily where? Is it in the RER or in the Golgi? Usually in the Golgi apparatus. Anything else? You mentioned packaging. Packaging occurs late in the process, and it's usually near the cytoplasmic face of the Golgi apparatus. So you're forming vesicles, okay, and packaging. But what else is occurring to this fairly early on? Somebody mentioned it, I think. This thing is going to be a straight rod? Okay, it's going to fold. But what happens during folding? What's one of the big things that happens during folding? What kind of bonding? Disulfide bonds form. Okay, disulfide bonds form. You make secondary and tertiary structures. Okay, so you start moving toward the end structure of the protein. And as it's moving, particularly through the Golgi, you may make modifications like carbohydrate addition. Starting in the rough endoplasmic reticulum, you're going to have some of these other capping modifications that change the charge in certain areas of the protein. What if it's a multi-subunit protein? Where do those subunits associate? Where would you expect them to associate?
In the cytosol.
In the cytosol? Okay. If they're going to be secreted, they're not going to be in the cytosol. So where would they begin to associate? So we told you about the gonadotropins, alpha and beta subunits, right? So at some point you have to have those alpha and beta subunits associate with one another. Where does that occur? Yeah.
So each subunit is folding within the endoplasmic reticulum, right?
Correct.
So ideally after that they have to be so the Golgi.
Okay. Probably within the Golgi apparatus, but there's nothing saying that multiple proteins can't be translated at the same time in the same stretch of the RER. Okay. So that association may begin to occur in the lumen of the rough endoplasmic reticulum.
Then how do you differentiate them as subunits if they're still already squished together?
Well, number one, there's this folding process that's occurring.
So they're folding separately, but they're still.
They're folding separately, and they begin to associate while they're there. What's going to make them associate more? It actually happens late in the process. During this packaging process that puts things into vesicles that are going to be ultimately be exocytosed. Okay. What do you think is going on there? Anything? Is it stasis? Are they stable? Okay. Those vesicles form in part because small molecules like water are being excluded. That increases the concentrations of whatever else is left behind, which tends to cause associations to occur. Okay. Now, so you've got folding, modifications occurring beginning in the rough endoplasmic reticulum lumen, continuing through the Golgi apparatus, and continuing on into these vesicle packages at the face of the Golgi, the extracellular face of the Golgi. So you've packaged these, you've got proteins. Do you think any of those modifications continue during the packaging process?
Yeah.
Yeah, they do. And in addition, you're going to have limited proteolysis that also occurs during movement through the RAR and into the Golgi apparatus. When we talk about insulin, you'll find out that there's a peptide called a C-peptide that's clipped out after the disulfide bridges have bridged and associated the other parts of the hormone. So there's proteolysis that can occur also as you go through the Golgi apparatus. And indeed, that continues on through the packaging. Those hormones that get to those vesicles aren't static. Okay? They are still undergoing modification. If they stay there long enough, they're going to be clipped. They're going to be modified. They're not going to be fresh, new hormone. They're going to be more aged hormone that's not as biologically active as the newest hormone made. And there's been good studies that were done in the late 60s and 70s that actually followed this process. And one of the things they found out was that under stimulus, pituitary cells and insulin-producing beta cells release vesicles containing the newest hormone that's been made first, prior to releasing older hormone that's been sitting around a while. And the biologically active hormone is released first rather than the older stuff that's been sitting around in the cell for a while. If you take time and measurements in women who are near the peak of their ovulatory cycle, when LH is released fast, what you find is the hormone that comes out first on the rising slope of the peak is more biologically active on a weight basis than the hormone that's released later. Okay. And it's all because of this processing system. Okay. All right. Have a good weekend.